Have you ever wondered what tells a towering oak to grow leaves and an ant colony to build a complex nest? It feels like magic, but it's science happening on a scale too small to see. Inside every living thing, from the smallest blade of grass to you, there is a hidden blueprint. It isn't a scroll or a file. It's a microscopic instruction manual inside tiny rooms we call cells. This manual guides growth, function, and life itself, consulted every second of every day. It is incredibly detailed, telling your eyes their color and your heart how to beat. It's a book of life written in a language all living things share, a 4-letter code scientists are learning to read. Think of life as the most complex construction project. The plan is DNA. Each cell holds nearly the same master plan, but different cells read different chapters. This journey needs no spaceship, just a microscope and curiosity. By the end, you will see how DNA, RNA, and proteins work together in a dance that powers every form of life. And this story is unfolding inside you right now. Molecular biology studies how life works at the level of molecules, the nuts and bolts inside cells. Not just cells as blobs, but the actual machinery, DNA, RNA, and proteins, and their relationships. In essence, DNA to RNA to protein. Understand this flow, and you understand life. DNA is a beautifully stable double helix, about 2 meters if stretched from one cell, tightly packed into the nucleus. Its backbone frames rungs made of 4 bases, A, T, C, G, whose sequence encodes information. Order matters like letters forming different words. Small DNA segments with specific instructions are called genes. All DNA is a vast library. Books are chromosomes. A gene is a single recipe for a task. A pairs with T, C with G. Know one side, you know the other. Crucial for accurate copying during cell division. A cell is a miniature city. Mitochondria as power plants, ribosomes as factories, highways, and a city hall, the nucleus. Proteins are the workhorses. They build, carry, signal, defend, and give traits from hair color to oxygen transport. DNA stays protected. Ribosomes are outside. The cell needs safe copies of instructions. This is where RNA helps. Orders go out, proteins are built. Cargoes move, energy flows, governed by the DNA code. Before a cell divides, it must copy its entire DNA accurately so each new cell has full instructions. Replication begins as the helix unzips. Special proteins break AT and CG connections. Each single strand becomes a template. DNA polymerases read letters and match complements to build new strands. The result? 2 identical molecules, each half old, half new. Proofread for extraordinary accuracy. Cells don't use all genes at once. To build one protein, first they copy one gene, transcription. Like writing a recipe on a note card, the cell makes an RNA copy using U instead of T. Proteins find a gene's start, open DNA locally, and RNA polymerase builds mRNA, which then leaves the nucleus. In the cytoplasm, ribosomes read mRNA 3 letters at a time, codons, each specifying an amino acid. AUG codes methionine, GGC codes glycine. tRNAs deliver amino acids to form a growing chain. A stop codon ends translation. The new protein folds into a shape to do its job. A mutation is a permanent change in DNA letters, A, T, C, or G, arising by chance or environment. Some slip past proofreading. Others come from UV or chemicals. Changes can swap, delete, or insert letters. Effects range from silent to subtle efficiency drops to major functional changes in proteins. Some mutations cause disease. In sickle cell anemia, a single letter change deforms red blood cells and impairs flow. But mutations also fuel evolution, occasionally creating beneficial traits that help species adapt. PCR, polymerase chain reaction, is a molecular photocopier for DNA. It turns tiny amounts of DNA into millions of copies for analysis, critical in forensics, diagnostics, and ancient DNA. By cycling temperatures with key ingredients, PCR repeatedly tricks DNA into copying itself in a tube. CRISPR is like molecular scissors plus a word processor, programmed to target a specific sequence. Cas9 cuts at the chosen spot. Repair can disable a gene or insert a new sequence to edit it. CRISPR helps reveal gene function and may fix genetic diseases, but its power demands careful ethical oversight. We toured DNA, RNA, and proteins, how cells copy and read the code, why mutations matter, and tools like PCR and CRISPR. Why should you care? Because this knowledge touches your health, food, and future. In medicine, we diagnose precisely, target faulty proteins, develop gene therapies, and design mRNA vaccines. In agriculture, we breed crops to be more nutritious, resilient, and sustainable. In the environment, engineered microbes help clean pollutants and restore ecosystems. Understanding our hidden molecular world connects us to all life. We share the same elegant machinery. Incredible complexity and efficiency arise from a simple four-letter code. The more we learn, the better we can heal, protect our planet, and solve once unsolvable problems. The tiny instruction book in our cells may be the most important story ever written. Our future depends on how wisely we read and use it.